Leonardo da Vinci left no children. That was the end of his bloodline. For 5 centuries, the statement seemed complete. Then researchers opened a trail of baptismal registers, tax declarations, marriage contracts, and notarial papers stretching from a Tuscan ancestor born in 1331 to men alive today. Among the artifacts was a 15th century letter written by Frosino di Ser Giovanni da Vinci, a relative from Leonardo's paternal family. Its folded paper had survived wars, floods, handling, and nearly 600 years of ordinary dust. In the fiber, scientists hoped, might remain something less ordinary, fragments of the Da Vinci Y chromosome. This is not a story about identifying or intruding upon living relatives. They cooperated privately with parts of the research, and their names and lives deserve protection. Nor are they Leonardo's direct descendants. Leonardo had no documented offspring. They descend to the same paternal family, mainly from his half-brothers, and that distinction matters. What the investigators built was a family tree spanning 21 generations and roughly 690 years. What genetic work later found among 6 living male-line relatives moved the investigation from ink toward biology. Then, in January 2026, researchers reported a possible genetic connection between the old letter and a red chalk drawing associated with Leonardo. Possible is the essential word. Remember the name Frosino, and remember paternal line called E1B1B. We will return to both when the paper record and the genetic trace finally meet. Stay with this. What the record can establish, and what it still cannot, is worth the time, because the most important discovery was not a gene for genius. It was that the family line history had declared finished had never actually disappeared. In the hills around Vinci, the official story begins with an unequal birth. Leonardo was born on the 15th of April, 1452, the son of Ser Piero, a rising Florentine notary, and a woman named Caterina. His parents were not married. Ser Piero entered the child into the Da Vinci household, while Caterina married another man. Leonardo grew up near his father's family, entered Andrea del Verrocchio's workshop, and eventually became the painter, engineer, anatomist, and observer whose notebooks seemed to contain several futures at once. He never married. No child can be securely documented as his. When he died in France in 1519, his property passed to pupils, servants, and relatives. His family name, in the modern imagination, became attached to one solitary genius rather than to a continuing household. Even the phrase Da Vinci was not originally a hereditary surname in the modern sense. It identified a connection to Vinci, that is the official account, and in its central facts it remained sound. The first crack appeared not in Leonardo's biography, but in the assumption built around it. A person can leave no children and still belong to a biological line that continues through siblings. Ser Piero fathered many other children. Leonardo had at least 22 half-brothers, and a branch descending from his half-brother Domenico carried the paternal line forward. In 2016, Alessandro Vezzosi, the founder of the Museo Ideal Leonardo da Vinci, and historian Agnes Sabato announced a reconstruction of the wider family. But early genealogy was not enough. Names repeated, spellings shifted, parish books contained gaps. One person once proposed as Leonardo's child turned out to be a different man with the same name. So the researchers kept going. Their later study, published in Human Evolution in 2021, traced a direct male line from Michel, born in 1331, through Leonardo's father and collateral branches to the present. It covered 21 generations, 5 branches, and from the 15th generation onward, more than 225 individuals. 14 living male relatives were identified at that stage. The paper did not produce Leonardo's genome. It produced something science needed first, a map of who could legitimately be compared with whom. And on that map, one fragile chain of fathers and sons became the route toward a chromosome Leonardo may once have carried. The investigation began in archives, not in a genetics laboratory. Vezzosi had spent decades studying Leonardo's world, while Sabato worked through the human landscape surrounding the famous name. They were not initially searching for a hidden ethnic identity or a biological explanation for genius. They were trying to correct a family record, checking ancestors, descendants of relatives, occupations, properties, marriages, and deaths. The genetic possibility emerged from that documentary reconstruction. This agenda-free starting point matters. A sensational theory can select only the evidence that flatters it. A genealogy must survive the dull things: repeated names, unpaid taxes, remarriages, missing pages, and the possibility that a father named in a record was not the biological father. Their first evidence track was therefore archival. Notarial lacks linked households. Church registers fixed baptisms, marriages, and burials. Property documents showed continuity across places and generations. The researchers corrected earlier errors and extended several branches into the living present. The result did not make the 14 men Leonardo's descendants. It made them paternal relatives descended from the wider Da Vinci family. The strongest objection is obvious. A documented father-to-son chain over nearly 7 centuries may record social paternity, not uninterrupted biological paternity. A single unrecorded adoption or non-paternity event could break the Y chromosome connection, while leaving every document apparently correct. This is a legitimate challenge. It cannot be dismissed. The research team acknowledged it directly. Their methodological response was not to trust one modern relative. It was to locate multiple living men belonging to separate documented branches and compare their Y chromosomes. If men whose branches separated generations ago share the same distinctive paternal signature, the probability that the archival line reflects biological continuity becomes much stronger. It does not make every link infallible. It creates an independent test of the paper trail. By 2025, the project reported that 6 cooperating male line relatives shared matching portions of the Y chromosome. This finding was announced alongside a new research volume, an ongoing analysis of burials associated with the family. It was an important advance, though not yet the same thing as recovering Leonardo's own authenticated DNA. The archive had found the living comparison group. The next problem was harder. Scientists still needed a sample that could be tied securely to Leonardo, or at least to a dead man unquestionably inside his paternal family. The search then moved from family names to microscopic residue. The Y chromosome passes mainly from father to son. It accumulates changes slowly enough that related paternal branches can retain recognizable markers across many generations. That makes it useful for testing the Da Vinci male line. It cannot reconstruct Leonardo's entire ancestry, and it tells us almost nothing about his mother's lineage. It is one narrow biological thread. Researchers pursued several possible sources. One was a family tomb at Santa Croce in Vinci, associated with Leonardo's grandfather Antonio and other relatives. Remains recovered there could connect modern men to earlier generations. Another was a grave in the chapel of St. Hubert at Amboise, marked only as containing remains presumed to be Leonardo's. That word, presumed, contains an entire historical disaster. Leonardo was buried in the collegiate church of St. Florentin in 1519. The church was later demolished after damage connected with the French Revolutionary era, and graves were disturbed. In 1863, Arsène Houssaye excavated the site and selected a partial skeleton near stone fragments bearing portions of Leonardo's name. Those bones were reinterred at St. Hubert. The identification was plausible, but never conclusive. The project therefore explored a third source: objects touched by the Da Vinci family. A non-destructive swab can collect minute biological traces from paper or chalk without cutting the artifact. But an old surface is an ecosystem. It can hold bacteria, fungi, plant material, animal glue, and human DNA from makers, owners, restorers, curators, and viewers. In January 2026, a preprint, not yet peer-reviewed, described DNA from objects associated with Leonardo's circle. The most discussed item was a red chalk image known as the Holy Child. Male-specific fragments recovered from several swabs were placed broadly within the E1b1 lineage, with support for E1b1b sublineages. Related signals appear in the letter written by Frosino, the paternal relative named at the beginning. The match was intriguing. It was not identification. The drawing's attribution to Leonardo is disputed. Even if he made it, another Tuscan man could have handled it. Sparse ancient DNA can also be distorted by contamination and laboratory limits. The preprint demonstrated that cultural objects may preserve useful biological traces. It did not prove that Leonardo's genome had been recovered, but now the second evidence track had reached the edge of the first. This is where the two tracks meet. The documentary evidence and the genetic record were pointing in the same direction, though not yet with equal certainty. The archive traced paternal relatives across 21 generations. Tests among 6 living men reportedly found a shared Y chromosome signature. Separate swabs from a family letter and an attributed Renaissance drawing produced male line fragments compatible with a broader paternal lineage found in Tuscany and elsewhere around the Mediterranean. Neither result alone can name Leonardo. Together, they define a testable route. Return now to Frosino. His letter matters not because it carries a secret confession, but because its writer occupies a documented position in the Da Vinci paternal family. If DNA from that paper can be distinguished from later handlers, compared with the 6 living relatives, and then matched to authenticated remains in the family tomb, 3 dates separated by centuries could be joined biologically. Only after that foundation exists could the presumed bones at Amboise, or securely attributed Leonardo object, be tested with real confidence. There is another layer the paternal search cannot solve. Leonardo's mother, Caterina, remains the great open branch. For generations, she was commonly described as a local Tuscan woman of modest status. In 2023, Leonardo scholar Carlo Vecchi publicized a Florentine emancipation document signed by Ser Piero and argued that Caterina had been an enslaved woman brought from the Black Sea or Caucasus region. The document is real and the argument is serious, but the identification remains debated. The name Caterina was common. Linking the emancipated woman to Leonardo's mother requires a chain of contextual inference, not a genetic match. Even a complete Y chromosome from Leonardo could not settle it, because that chromosome came through Ser Piero. This unexpected limitation changes the meaning of the project. Science may recover paternal signature while leaving half of Leonardo's immediate ancestry in shadow. A mitochondrial profile, passed through the maternal line, would require Leonardo's authenticated remains or securely traced maternal relative. Neither is presently available with comparable confidence. The living relatives therefore carry part of the answer, not the whole man. Their DNA may authenticate a skeleton or reject a false attribution. It cannot explain the Mona Lisa, derive creativity from a haplogroup, or turn cultural achievement into heredity. There is one more reason the record became so uneven. It lies in the social machinery of Renaissance Florence. Renaissance archives preserved lineage because lineage controlled power. Florentine notaries recorded contracts, dowries, inheritance, legitimacy, property, and civic standing. Ser Piero belonged to that professional world. His male ancestry was valuable because offices and estates moved through recognized family structures. Caterina's identity was recorded far less fully because a young unmarried mother, possibly of low status, and perhaps enslaved, held a different legal and social position. The silence was not necessarily a coordinated concealment. It was structural. The archive asked detailed questions about men whose property and professions mattered to institutions, while reducing many women, servants, migrants, and enslaved people to a given name and a relationship. Modern researchers inherit that imbalance. The father's line becomes a sequence. The mother becomes a dispute. Leonardo's case is not unique in its structure. It is unique only in the prominence of the name attached to it. Early modern Europe produced countless family histories in which elite male lines can be followed through deeds and registers while maternal origins dissolve after 1 or 2 generations. Genealogists call the disappearance of women's birth names and origins a form of archival invisibility. Geneticists encounter a related problem. A pedigree may look continuous while biological paternity remains uncertain. The Da Vinci Project addresses one weakness with the other. Documents tell scientists which branches to sample. DNA can test whether separated branches really share a paternal ancestor. Archaeology can place bones in a family tomb. Material science can determine how an object was made and handled. None of these disciplines is sovereign. Their value appears when they constrain one another. The living present matters here. The men who supplied samples are not museum specimens and not vessels of Leonardo's genius. They are farmers, artisans, office workers, and other private citizens whose connection to a celebrated family became scientifically useful through an accident of inheritance. Their shared Y chromosome does not make them lesser copies of Leonardo. It simply preserves one paternal signal that may help resolve a historical identification. And Leonardo's achievements require no genetic rescue. The Last Supper, the anatomical sheets, the studies of water, flight, optics, and geology came from a life of observation, training, opportunity, discipline, frustration, and relentless curiosity. A genome could suggest traits or health risks. It could never contain the meaning of the work. The pattern is therefore larger than one famous bloodline. Archives decide whose continuity is visible. Biology sometimes restores a connection, but also exposes what the archive never cared to preserve. At Amboise, the stone above the grave still refuses certainty. It does not say that Leonardo lies there. It says the remains are presumed to be his. That restraint is the most honest inscription in the story. The family tree is strongly documented. The shared paternal signal among living relatives is a promising biological anchor. The family tomb may supply an older bridge. The Holy Child and Frosino letter offer a provocative experiment. Yet the final comparison, the one that turns compatibility into identity, has not been completed in a form accepted beyond reasonable scientific dispute. If it is completed, the first achievement may be simple: confirming or excluding the bones at St. Hubert. The same reference profile could help evaluate disputed objects when a credible chain of custody exists. Researchers may learn something about Leonardo's appearance, health, or sensory biology, but those claims would demand unusual caution. Complex abilities do not reside in a single switch. Genes interact with development, nutrition, illness, education, culture, and chance. And ancestry is not destiny. The paternal haplogroup E1b1b, if ultimately confirmed for Leonardo, would place one narrow male line within a lineage found across broad parts of Europe, North Africa, the Near East, and the Mediterranean world. It would not assign Leonardo a modern national identity, prove a dramatic migration by itself, or settle Caterina's origin. A haplogroup is a branch on one tree inside a forest of ancestors. Travel back 21 generations, and every person has an enormous theoretical number of ancestral positions, many occupied by the same people because family trees overlap. One surviving Y chromosome represents only one path through them. So what did the investigation truly change? It replaced the image of a biological dead end with a living network. It showed that Leonardo's story did not begin with an isolated miracle in 1452. Behind him stood Tuscan households, working people, notaries, women whose surnames vanished, and perhaps migrants whose journey is still contested. Beside him stood half-brothers whose descendants carried a molecular fragment forward without knowing what science might one day ask of it. Now return to the letter. Frosino folded it for an ordinary reason almost 6 centuries ago. He could not have known that its dust might be compared with living blood, excavated bone, and chalk touched in a Renaissance workshop. The paper has survived. The chromosome may have survived. But the human story remains larger than both. And when the science is finally finished, one question will still be waiting. How much of a life can blood remember?